So I'm going to kick off the introduction for the DES Blackleg project. So the ultimate aim of this research is to create a decision support tool for potato blackleg disease. So an overview of the work can be seen here. I directly work on work package 3 in the bright red, and then Mansi and How work on work package 3 and work package 1. So in my area, we're going to use microbial analysis tools to understand blackleg disease and determine potential control measures. So the team at University of Glasgow includes Professor Barbara Mabel, who's an evolutionary geneticist, Dr. Umar Ijaz, whose expertise is in integrated omics analysis, Dr. Joel Milner, whose expertise is in plant microbe interactions, and we also have Professor Dan Walker with a specialty in bacteriocins. And then there's me, the postdoc on the project. So I joined in January. So just to give a little bit of background about myself. So I'm an environmental microbiologist with a BSc and PhD in microbiology. And I've always been drawn towards understanding complex microbial communities that have a relevance for society. So my PhD was focused on low temperature anaerobic digestion for sustainable waste treatment. And then I moved to Glasgow in 2016, working on a Frontiers in Engineering grant and a Global Challenges grant in collaboration with researchers in Thailand, with the aim of developing novel tools to understand anaerobic microorganisms and to understand the microbes in solar septic tank systems. I was drawn to the Blackleg project as this research is cross-disciplinary, involving multiple collaborations to understand a real-world challenge that can be solved by controlling or manipulating specific microbial populations. And I think this can really only be achieved by understanding the microbe to microbe relationships and also the dynamic changes associated with the environment. So the vegetable production industry suffers high monetary losses from pest infestations, as I guess everybody here knows. And one of the most popular vegetables in the UK, the potato, has estimated losses of over 50 million per year in the UK attributed to blackleg disease. So in this disease, or also called soft rot, the bacteria produce huge amounts of extracellular enzymes, which lead to decay symptoms or breakdown of the cellulosic plant structures. And it's largely attributed to Pectobacterium species. And Pectobacterium atrosepticum is traditionally believed to be one of the more important causative agents for blackleg in temperate climates and the dominant agent in Scotland. So it's a pathogen belonging to the Enterobacteriaceae family, and these are facultative anaerobes, species that are typified by the production of this pectinolytic enzymes during plant infection. The species is difficult to control as it's likely part of the natural soil microbiome, and strategy for control of potato soft rot and blackleg are limited. So no blackleg resistant potato cultivars are available. And one of the main approaches is to reject infected plants. Seed certification schemes are also employed, but the success can vary. So there's a pressing need for innovation in how to control Pectobacterium infection in crops. So our strategy is to characterize the microbial community from both the root and soil from various field trials. Thus, we'll be able to link with agricultural practices such as cover crops, irrigation, and nematode treatment. We'll then explore one potential mechanism that could provide a targeted approach to kill Pectobacterium atrosepticum, and that is the use of bacteriocins. So bacteria weaponize bacteriocins, which are proteinaceous compounds with a narrow killing spectrum, to kill their enemy or closely related species so that they can take their place in the niche. And so in turn, we can weaponize bacteriocins against these plant pathogens without impacting the wider soil microbial community.
I have gained molecular expertise in molecular biology, and I have decoded a novel role of phosphatidylinositol 4 kinase gene from Nicotiana benthamiana as a negative regulator of plant immunity against Gemini viruses, which are single-stranded DNA plant viruses. Next. Yeah. We found the association of the host factor, that is PI4 kinase from Nicotiana benthamiana, with the viral replication initiator protein, which is involved in replication. The association was found to be direct and taking place into the nucleus. Next. Then we characterize this host factor from Nicotiana benthamiana, the solanaceous plant, and found that this protein is a functional protein with ATPase activity. But as opposed to conventional lipid kinases, it lacked lipid kinase activity, but it has come to have a novel protein kinase activity. Next. Then we performed the silencing of this host factor in Nicotiana benthamiana and found that upon silencing of the gene, there is drastic reduction of viral symptoms, as well as the onset of the symptoms was also delayed. So we concluded that this PI4 kinase is a novel host susceptible factor, which is exploited by the virus for its own pathogenesis. So my experience of working with the solanaceous plants like Nicotiana benthamiana, Nicotiana tabacum, and Capsicum enum will help me with the current project, which is on potato. Next. Now coming on to the current project, as we know that plants influence its rhizosphere by secreting the root exudate. The components of the root exudate, it can contribute to symbiosis, or some of the factors may also act as defensive agents against microorganisms. Next. So our aim in the current study is to isolate the root exudate from the potato plants, which are subjected to different conditions like irrigation or those grown after the cover crop, and to identify the key markers in the root exudate, which can be utilized as a tool to fight off the blackleg disease. And my broad expertise in molecular biology will help me in characterizing the the markers of the root exudate to fight off the blackleg disease. Yeah, thank you.